Yes. Okay. Hi, everybody. I'm Mirae Kim, and I'm from RegSys, and I work with Dr. Vicky Yao. And today I'll be talking about ontology-aware prediction of tissue-specific DNA methylation. So, as Emily has said previously in the previous presentation, DNA methylation is part of a family of processes called epigenetics, and it is what lets us modify gene expression without modifying the actual DNA. And DNA methylation, specifically speaking, acts by adding a methyl group to a site called CpG site, C followed by a G. And then by doing so, it carries out the main function of transcription regulation. And it does so by methylating these regions, most prevalently those promoter regions that are in front of genes that it wants to inhibit. And by doing so, it carries out different gene expression modifications, meaning that some of those genes may or may not be expressed depending on if the methylation is there. And because of this, and because the fact that we have so many varying tissues with so many different characteristics and functionality across the body, even though they all have the same DNA, there appears these tissue-specific regions of depending on those methylation markers. And also an interesting fact about methylation, as mentioned before, is that it actually is adaptable to the environment or the lifestyle choices that a person might make, along with those habits and such as smoking, and then but also those changes could be happening due to tissue specificity or any stable stimuli that could be happening to that tissue. So it's an interesting difference to look at those that are short-term stimuli like environment versus a long-term stable stimuli like tissue-specific. And on top of that, DNA methylation and tissue specificity, which was also mentioned before, has been studied before, where in terms of deconvolution of methylation markers, or in terms of validation of tissue-specific DNA methylation in non-invasive or invasive cancers or cancer subtypes. And also mentioned before, DNA methylation age clocks has been on the rise, and they have been found to be useful in terms of predicting methylation age, which then can have a strong relationship with chronological age where the samples are from. And due to this, with the strong relationship that it finds, it actually has an interesting application further on where the deviation of the predicted age from the chronological age can be looked at. And depending on what the conditions of the person are, there could be some sort of an age accelerating or age decelerating condition, which is interesting to look at just by looking at the methylation levels. And then at this point, then we know that the aging clocks have been impactful, and these are actually able to carry out this function with a couple hundred probes. And because of that, and they're also multi-tissue. So because of that, the question that pops up in our head was that can something similar in concept or parallel in concept be done with tissue-specific DNA methylation? And can we have a normal tissue classifier using DNA methylation, which then can be looked at for deviations in tissue-specific methylation levels if there is a tissue-specific or multi-tissue disease that is going on? However, current state of classifiers are very limited to those that are in the training set. So those tissue labels, which then need to be collected into a compendium, and then it needs to be learned by a classifier. However, as human data goes, having all the tissue samples from everybody that covers all the variations is not the easiest problem. And to kind of mediate with that, we use Uberon tissue ontology, which is a structured relationship between tissue labels, where it gives you a directed acyclic graph, a DAG, and all the nodes in that graph are tissues, and then all the edges indicate physiological or proximal relationship between those tissues. And then we specifically use the edge types of ISA and part of, which can be explained using this very zoomed in version of the ontology, especially in the blood cell types. So in this example, leukocyte and peripheral blood cell are connected, so which means they're related in some sort of a way in terms of their function or proximal location. And then the edge type is either an ISA or part of, so it would mean that leukocyte is a subtype of peripheral blood cell. So all of these parental and children relationships actually indicate the physiological function or relativeness of those tissues, which then can be used as external information in terms of which tissues are related to which. And then it can be also used for maybe gaining an idea or at least tiny bit of an insight in terms of tissues that are not within the training data set. And then, so using that and knowing that we have some sort of a systematic relationship and there exists some sort of tissue-specific DNA methylation, this question is the one that we're trying to answer, where can we use methylation and structured relationship between tissues from the ontology to predict robust tissue-specific DNA methylation? And our general framework of ontology-aware prediction of tissue-specific DNA methylation is like this, and then we'll start off with our data. Our data is manually curated multi-tissue human methylation data set from the GEO, gathered using platform of Illumina Human Methylation 450K. And then we further narrow down that data set using a couple criteria. The first being there's no known disease at the time of measurement, so normal tissue only. And then additionally, we want no drug treatments or external stimuli of any kind at that point, which then narrows the set down to about 3,104 samples from 114 studies over 47 tissues. However, we also wanted to focus on the robustness of the training set that the classifier is going to see. So we then further narrow down the tissue labels to those colored in blue, which have multiple tissues, source of studies for each of the tissues, rather than having just one study for that tissue label. And then the next part, we then feed that data set into our feature selection, which then for feature selection, we use our collaborators' algorithm, mini-patch learning. Mini-patch learning in simple terms works like this, where a mini-patch of random sample subset and a random CPG subset is picked at each iteration. And then that mini-patch is put through a classification. It's a very simple classification resulting in feature importance. And then the CPGs that were in that mini patch are divided into those that had high feature importance and then those that had low feature importance, where those that had high feature importance are actually exploited by getting picked again and again for those of the next iteration of the mini patches. And then those with low feature importance are actually replaced during the exploration algorithm, where the rest of the CPGs that did not get randomly picked during this iteration are replacing that. And then by iteratively going through that process, it results in overall selection frequency, where due to the exploitation and exploration algorithm, the selection frequency is heavily tied inherently to how useful that CPG probe was throughout many of the mini patches throughout the entire data set, meaning its versatility and the measure that we can have as how important that feature is to the entire data set. Another way to very naturally think of feature selection in terms of, especially in DNA methylation, would be differential methylation. So I did actually perform differential methylation, one versus rest, on all of the tissue types that we do have on hand. And then I sorted the tissues by their ontological relevance, meaning like all the blood cell types are together, all those that carry out similar functions are all sorted together. And then I picked out top 10 differentially methylated probes from each of those tissue and then colored them by fold change. By looking at the fold change and the patterns that are happening, we can actually already see that there's some sort of structure going on between those that are relevant, meaning that there's some similar behaviors in terms of the methylation probes that are being methylated or not being methylated across those tissues that are relevant to each other. However, upon running time analysis between mini-patch and differential methylation, we observed that mini-patch with its multi-tissue capability, along with its efficient algorithm, is actually much faster than carrying out differential methylation. So then we proceeded with differential mini-patch learning. And with mini-patch learning and the overall selection frequency that it outputs, one of the very important hyperparameter is the selection frequency threshold, which means we pick a threshold and the probes that are over that threshold are the ones that we pick for the training and testing of the set. And with this, we ran a threefold cross-validation, which then ended up in 0.45 as our threshold, resulting in 741 probes out of 473,000 probes from the platform. And then we also made sure that the variations that were in the original set that were defining the tissue specificity from all of the 473,000 probes were not lost by doing this feature selection. So we validated by looking at PCA. And then from that moment on, then we used the 741 probes into our ontology-aware multi-label classification. In this case specifically, tissue labels were still at this point single annotated labels that were from the GEO. However, we wanted the classifier to be able to learn information just past just the labels themselves, where they are aware of the relationships in the ontology. So in this case, the way that we decided to do it was label propagation according to the ontology. Simply said, because of the relationships that exist in the ontology and the edges that we have chosen to make it work, being an inclusive is-a or a part-of relationship where the child is in one way part of the parent, we wanted to make sure that that was being reflected into the information that the classifier was seeing. So we propagated, for example, a sample of islet of Langerhans would then be propagated into a multi-label set of exocrine gland, pancreas, and islet of Langerhans, which would indicate that the sample should, in one way or another, in one resolution or another, should carry some information that is related to those three of the labels. And by using that multi-label set for every single sample, we put it into a multi-label SVM, and we were able to actually achieve high cost performance regardless of the level of the ontology that it saw. And all of the 26 tissue labels that we do have on hand at least has a minimum AUC of 0.9, with one exception of leukocyte, which is very notable, and which then can be also looked at for what kind of predicted labels that it has if it wasn't leukocyte itself. So we created this multi-label version of confusion matrix, where at this point, because each single label could have many of the correct answers, our main point, and when we're looking at this confusion matrix, is not to look at the diagonals itself. Actually, then we want to look at what kind of label set that it's predicting at once, rather than only looking at the diagonal, which is the true single label. And then it can be easily said when we look at leukocyte. So leukocyte, as we can see, is actually not doing so well in terms of its getting its correct single label. However, it is still predicting its parent node blood correctly. Actually, across the blood family, it is predicting blood correctly overall, meaning the classifier may not be learning individual labels so well. However, it is learning the general characteristics that are related to blood and across the different levels of the ontology. And then there are actually labels that it's getting all of the parental nodes and its children nodes correctly with high performance, which then gives us an idea that it is actually able to learn different resolutions of the methylation markers across the different levels of the ontology. And with that said, we also want to see if this performance carries out through those that were unseen during training, which was our motivation. And so we perform zero-shot learning with this classifier. And zero-shot learning classifications are made on those that were colored gray in the beginning when it was filtered out due to it not having enough samples or not having enough studies. So these labels are not part of the predictable labels that we're trained on. And because of that, we wanted to see if the relevant ontology would increase if we added these in. And indeed it does. Actually, the zero-shot learning labels are all across the ontology, which are called filter samples with red outline. And by looking at them, they are distributed regardless of where the learned samples are from, where the learned samples are colored in orange. And because the learned samples are the only predictable label set the classifier knows, it is actually not able to classify those that it doesn't know because we didn't give it to them. So the metric that we found was a node-to-node distance between the predicted set and the true node on the relevant ontology shown before. And how it's calculated is that because it is only able to predict the closest node, theoretically, to the filter sample, we calculate the sample-to-sample distance from the predicted node to the filter sample. And then we also compare random distance from a random node in the ontology to the filter sample. Then we have two distributions of samples of two different sets, which then we compare if the distribution of sample distances are significantly closer than those of the random distances. And in our case, of the labels that we had on hand, we have about half of them that are statistically significantly closer than those of the random distributions. And then if we look at the labels, they're of gastric gland, trachea, bronchus, stomach. And then so they have functional similarities within each other of those ones that they performed well in. So this phenomenon is actually way easier to look at if we look at it in terms of system-based nodes. So system-based nodes are functionally or proximally related, or both. And because of that, they have some similar methylation markers due to their shared functional characteristics. And in this case, if we group them together, then of these systems, we have statistically significant closer distance than the random distribution in digestive, respiratory, nervous, and hematopoietic and cardiovascular. And also these are the ones that we do have more coverage of the system in our training set, showing how important the data availability is in terms of performance. And in summary, we have a manually created normal tissue data compendium with 3,104 samples. We utilize tissue specificity of DNA methylation and tissue ontology to introduce an ontology-aware learning and prediction. Efficient multi-tissue feature selection using mini-patch learning is performed. And then we have overall good classification performance along with ontologically intuitive predictions. And we also extend to unseen label predictions using zero-shot learning, measured by proximity, by node-to-node distance in the ontology. And as our future work, we want to look at then the disease tissue drift from the normal tissue classifier that we have on hand. And then we also want to apply it to cancer of unknown primary prediction, which are essentially the metastasis of a cancer might have similar methylation markers to the primary, and then that could be predicted using a normal tissue marker, which has the tissue-specific methylations learned. And then we also want to investigate tissue specificity with additional resolution, where some annotations might not be available to the resolution that we need, or some of the annotations could be more detailed, and that could be learned, or that could be picked up by the classifier that was trained with more resolution. And then we also have tissue-specific weights to look at tissue-specific CPG sites, which then it could be behaving differently across more of the tissues if we have them on hand. And I want to thank all the members of my lab and our collaborator at the Columbia University, and I'm happy to take any questions.
Nice talk. You know, if you ask different people to define a leukocyte, you get different answers. And I think that could be reflected in how those leukocytes are being isolated to generate the methylation data. So I'm wondering if that could impact the performance of your models on that population.
Yeah, the point about the more in-depth tissue annotations actually was stemming from the leukocyte and its identity in the blood family. Actually, all the different ontologies also have different edges between the blood cell types and where they are located and all of that. So yes, I think it really could be the fact that definition of leukocyte is ambiguous throughout the data sets.
And that's how the samples are isolated experimentally.
Yeah.
Any other questions?
Hi. Yeah,
Hi.
I had a couple of questions about your prediction on unseen labels.
Mm-hmm.
First is just like a clarification. I wasn't sure how some of those labels, if they weren't present, are getting a minimum distance of 0. If like the true label isn't in the known labels.
Yeah, they were all normalized. The minimum distance was subtracted from all the distances. Yeah.
And then the actual main question is, do you have some metric you can look at to sort of say what you believe is like a true prediction for a cell of this class, and what you think this is an unseen label that's close to this label, that's, you know, this is the closest cell type. You know what I mean? Like some sort of uncertainty to say, you know, this is a lung sample that we're confident is lung, or this is something we think is similar to lung, but probably not actually lung.
I think in our case, the one that we found most useful was the distance on the ontology. It could be different depending on what physiological meaning that you're looking for, as in terms of metric. But I think it could be something like overlap in cell types that exist if it's a 2 different location in the body. But so far in our case, it was the node-to-node distance on the ontology.
I have a quick question.
Okay.
I think nice talk, by the way.
Thank you.
So I was wondering, how did you distinguish what kind of relations to use, you know, ISA versus RDOC if you were building the ontology?
Oh, there are other relationships, but they're not all inclusion-based. So it's not always like a one-way relationship between parent and child, and we wanted it to be a DAG specifically, so that it would carry a one-way relationship where the parent includes the child.
So
Yeah.
that would be ISA?
is a and a part of. Yeah.